In this video, I'm going to be talking about one of the most sinister and secretive weapons in cancer biology. A weapon that is quite sweet, but very dangerous. And we're talking about glycogen, AKA storage sugar. I've been focusing a lot on cancer. I've been focusing a lot about burning sugar via mitochondria and how that's related to both cancers as well as long COVID and post-vaccination syndrome or COVID-19 related injuries. And today we're going to discuss very interesting latest information, how cancers, specific type of cancers, can load up on sugar and use it as a weapon. My name is Dr. Rashik of Merogenomics. Let's get started with this. First of all, then, what's glycogen? As I mentioned, think of glycogen like a bush with lots of branches, but it's all made of sugar. It's a polymer. Whenever you heard the word polymer, it means chemicals are being linked together. In this case, the chemicals we're talking about are sugar molecules. And many cells store that for fuel. Good example are your muscles, your heart, because obviously muscles and your heart require lots and lots of energy. Brain requires tons of sugar for processing. Bulk of sugar in your body will be consumed by your brain. So brain can also store sugar like that. Liver is another example. And it turns out that so can cancers. So how does it help cancers? There's numerous ways in prior episodes dedicated to both how this technique used by cancers, how cancers burn sugar. I've discussed how you could try to counteract that with interventions, as well as I did a recently video on the very first clinical trial for post-vaccination syndrome individuals, and I'd explain the mechanics of it. But in essence, cancers can burn sugar without air, without oxygen. This allows cancers to survive in a hypoxic environment as well. So even when there is no oxygen being delivered, and that's important for cancers because sometimes they might not have a blood network to deliver everything they need, all the nutrients and so on. And burning sugar without oxygen, which is a hallmark of dysfunctional mitochondria, we can do this naturally, but not for very long, and then we shut down. Cancers can do it. Certain cancers can do it for periods of time, get nutrients as a consequence they need, and allow them to survive without necessarily oxygen being delivered anyway. On top of that, though, the metabolic components, because of this process, they promote angiogenesis. They stimulate angiogenesis. Angiogenesis is a fancy schmancy scientific word for promoting development or build-up of new blood vessels. So as the cancer grows, it eventually might need that supply. So that's another one. And the last one, the most sinister one, glycogen via sugar can be used to produce acidic environment around cancers. This acidic environment can start destroying what is referred to as extracellular matrix. Think of extracellular matrix as a skeletons, like a skeleton outside that shields cancers from blood vessels that can be destroyed. And this starts helping cancer gain access to blood vessels so that they can start metastasizing to other areas of the body. And the more sugar cancers have that they store, the more likely they're going to be prone for metastasis. So cancer cells can have literally like a tripling going from one area of the body to another, and glycogen could be helping with that process. So the more dangerous cancers, they will be basically hoarding sugar. And certain cancers are more likely to metastasize to other type of organs. This is in science referred to as organotropism. So an example is those sugar-prone cancers that use a lot of sugar, are known, for example, to be able to metastasize to your brain. So goes from one specific organ to another, perhaps because brain uses so much sugar as well. So it's a good location for such type of cancers, but you can see how dangerous glycogen could be for cancer development. And remember, for the dangerous type of cancer development, such as the one that metastasize, and remember, most people don't get killed by the original type of cancer. Most of the time, it's the metastatic stage of cancer that is the really deadly component to cancer patients. Okay, then, so that's a little bit of background on what glycogen is. Glycogen is being built by specific enzymes. One of them is glycogen synthase, so it synthesizes it, it makes sense. It's broken down by glycogen phosphorylase, I believe, and it's known that if you have more of that enzyme in your tissue, it's bad prognosis. All right, so this is your preamble. So some cancers are hoarders of sugar. Now, in these videos that I did previously, based on proposed mechanism of how you could attempt to fight cancer, taking advantage of this known mechanism, because this is very well established. I was mentioning, oh, all cancers do it, and then a bunch of readers mentioned, no, Mikolai, not all cancers are like this. So dependent on sugar, some are not. So it's usually the viewers, not the readers. It's the viewers that point out extra information to me, and I started studying this further. So indeed, not all cancers, but some cancers are far more prone to this type of mechanism of using sugar and storing sugar, and these are breast, kidney, ovarian, colorectal, and now we're going to talk about lung cancers as well. So this brings me now to the first study I want to talk about. And by the way, I'm going to link it all as well to mRNA vaccines at the end. What happens when you mix cancer cells and mRNA vaccines? So stay tuned for that as well. This will be the last study I'll mention. All right, then let's talk about the first study I want to tell you about in this video. What the authors wanted to do is they wanted to study the aggressive type of lung cancer called lung adenocarcinoma type of cancer. It's known to be aggressive, and if you have this type of cancer, such an individual usually has poor prognosis. And they wanted to look at that glycogen storage levels in these cancers. And they did something very, very interesting. And I want to tell you because of the technology that is now available. So what these authors do is they employ this technique called multi-mass spectrometry. So what is that? You, oh, I just swallowed the bag, I think. Oh well, bon appetit. You take cancer cells and you slice them super thinly. Imagine like slicing deli meat, but it's super, super thin. Then you put it, let's say, on your slide. You coat it with specific chemical matrix, and then you shine it with a laser on it. And this laser, because of its power, it allows to produce specific type of ions. What are ions? Ions are atoms that have electric charge. And they have this electric charge because they either lost an electron, meaning they are more positive. Another way you can call it is they are oxidized. And I'm mentioning this now because this will be important for later as well. Or they can gain an electron. So now they're going to be more negatively charged, and such ions are called reduced. And this technique can detect that. And these authors use this technique specifically to look for glycogen. And they would take a slice of cancer tissue and they would shine a laser at a tiny component of that slice and then move to the next. So they would do like a grid. So think of like pixels on TV, and they moved across the entire sample, and this allowed them to not only detect glycogen in the cancer sample, but it allows them to map exactly where all the glycogen is in the sample. we are entering meadows and so pretty, lots and lots of flowers, very beautiful. I hope you get the view. So not only do you see what's in there, you can now map it exactly, just like you would on TV with pixels, and that's exactly what they did. And the authors have this very cool figure where they show this. So let's show you this figure. So, and they use different colors, just like you would do on TV, to demonstrate different amounts or quantity of glycogen in a tissue. So on the very left-hand side of this one image, you see it's a normal tissue, and you can see in green and yellow, those are large quantities of glycogen, but you can also see white spaces. There is no glycogen there at all. So some tissues will load up on glycogen. Then they compare it with non-aggressive type of lung cancer. That's the one in the middle. And you can see lots of pink and purple. And this designates presence of glycogen, but at low levels, okay? And then look at the very right-hand side. This is the type of cancer cells that they wanted to study. These are the lung adenocarcinoma cell lines. And you can see lots of green, lots of yellow, and even red, which is the maximum amount of glycogen. And you can see these cells were completely loaded up with glycogen, tons of glycogen in there. And basically, these cells are hoarding sugar a lot. And the question is why? And we're gonna get to that. So they did this for lots of different samples amongst the different patients. And before we move on to more data from them, let me give you a little bit of background. So whenever a doctor, oncologist says, hey, this cancer cell line is well differentiated, it doesn't mean that it has a good taste in music. It means that, and this is a bit paradoxical, it means that the cancer cell looks a lot similar to normal cells, which you would expect from normal cells. So that's well differentiated. So well differentiated means that the cancer cells look very, very similar to what you expect from normal healthy cells to look like. So they're not too crazy. Now go to the poorly differentiated, and that's completely different definition. Hey guys. And at that point, it means that these cancer cells are super chaotic. They don't look anything like normal cells. They're just abnormal monsters at that point. So that's what poorly differentiated means. And there's also middle ground. All right, so then why do I mention this? Because they quantified the cancers of their patients, of these lung cancer patients, into well-differentiated versus poorly differentiated. And poorly differentiated cancer cells typically means poor prognosis, typically means cancer cells are already there. It's just not a good thing for such an individual. And when they looked at the glycogen storage, they clearly saw that the more poorly differentiated the cancer cell was, or in other words, the more abnormal cancer cell was from what you would expect it to look like in comparison to normal cell, they had more and more glycogen stored. So more of that sugar storage for future use. Again, check out my past videos to explain why cancers use sugar and their prolific uses of sugar, but they don't metabolize sugar the same way that you and I, normal healthy cells would. So that's the difference here. Okay, so there's that. And unfortunately, in essence, again, it shows you that the worse the cancers, these lung cancers were, the more sugar they were hoarding, as if they were preparing for war. And that's unfortunately exactly what it was, because these guys also showed us these survival curves. And you can clearly see in the survival curves that those who had more glycogen in their cancer samples did not survive as long as those who had less glycogen in their cancer samples. You can clearly see that people whose cancer cell lines had more sugar in them, more glycogen, they were dying faster. So that's the unfortunate part. But they did something else. Then they moved to looking at mice. And what they did is they did these experiments using mice where they fed mice Western style diet, so full of sugar, the typical Western diet that is just full of sugar. And they fed them for, can't remember, maybe a couple of weeks. And what they showed subsequently is that their lung cells loaded up with glycogen. They were full of glycogen thanks to this diet. Then they induced cancer, and they saw that once these cells, lung cells, were induced with cancer, the cancer was progressing worse than if you didn't have that prior load up with sugar. So, but here's the kicker. If you had your cells loaded up with sugar and they induced with cancer and they removed sugar at that point, it was too late. It was too late because even if you reduce cancer, reduce glycogen or sugar food source, cancer was still managing without it. It managed to still create its own glycogen and it was using the stores. So in essence, it was the diet prior to even cancer taking place that determined eventually how dangerous the cancer was. In other words, fast food equals future of fast cancers and worse outcomes. And obviously that's a plot twist no one was hoping to ever see. And the author specifically mentioned in this article that perhaps we should be looking at correlation between diets and cancers. And that should be investigated in detail to what degree our diets could be driving negative outcomes of cancers in the future. So that was the first paper. All right, so now let's go to the next paper I want to tell you. And this is now a paper from Poland. I think this was maybe published in 2023. I really like reading Polish science literature. Yes, I'm biased being born in Poland, obviously, but it's also because Poland has this phobia. I'm exaggerating, and it's a cliche, but Poland has this phobia of its culture being erased because in like the historic subconsciousness, there were attempts to do that in the past, and therefore, one of the best methods believed in the population to counter that effect if somebody attempts to erase your culture is to educate yourself prolifically. And as a consequence, Poland has had very high standard of education. So what do I mean? Let me give you an example. When I was in elementary school in Poland, I was already learning information that in Canada, later on, I would learn at university level. It was absolutely ridiculous. It was just crazy that these kids who wanted to play had to learn such crazy, complicated concepts so early on in life. And as a consequence, I like reading Polish science papers. They are complex, sophisticated, and yeah, and like I said, and I'm biased. So you're not supposed to be biased as a scientist, but it's also impossible not to be biased. All right, so what did those guys do? And why did I even want to bring it up in this particular video is because They took the same type of cancer cells, the lung adenocarcinoma type of cancers, just like the ones that I told you were being studied in that paper I just discussed with you a second ago. And now this wasn't from a patient. Well, I mean, it was at some point. This is a immortal cancer cell line. So you can study it all over the world. You can get your hands on that cancer cell line. So this is not exactly what mimics biology in the body. So keep that in mind. But what they did is they took that cell line and they wanted to see, hey, what happens if we expose it to mRNA Pfizer vaccine, COVID-19 vaccine? And they did that and exposed it, and they wanted to measure different things or one specific thing. So now and they measured it for, say, an hour up to several days, and then they even measured outcomes 2 weeks after the initial exposure of the cell line to the vaccine. But what did they do? They use another technique, laser technique, that is called Raman. It's either, I can't remember, it's spectrometry or something like that, but it's Raman. Ironically, I should know because I worked with that tech before when I was involved in development of nanorobotic constructs. But it's either spectrometry or something like that. But it's Raman. Think of it like imaging, but what it does, it's the laser shines onto your biological material, and the laser will scatter differently depending what biological material it encounters. And as a consequence, this difference in scattering that occurs due to atoms that make up the biological material that is being hit by the laser, you will be able to detect that difference. And by the way, speaking of nanorobots, I will start a series, hopefully on this trail, discussing what kind of nanorobots are being used in medicine right now. As I just mentioned, I've worked with that before, so I have a bit of background in it, and it's quite fascinating. It's crazy how this field is moving in terms of what kind of microscopic robots are being designed. Ooh, let's see if you can spot the lake there. All right, so stay tuned for that. So that's Raman, and they have this super cool image that these authors showed where they did the same thing. They took a slice of these cancer cell lines, they put them on a slide, and they shined that laser, and they did the same thing like the authors of the previous paper where they pixelated it, meaning they went into tiny area, and they moved on to the next area, and so on. They created a grid. And so once again, it allows you to not just detect what's there, but detect where it is all over the slide. And they show this super cool image showing how they use this technology to map cellular contents, but like large scale organelles. So we can see in that image, they showed with this technology where the nucleus is. So nucleus, they highlighted with red color. So again, they used the pixelation like TV where they added colors to it. You can see nucleus almost takes up all the space inside cell, which is really neat. Around that, you can see mitochondria. I think that was in pink. You can see how much mitochondria there is. So imagine, we need mitochondria for proper function. Imagine having these mitochondria no longer working properly for you. And therefore, why shutting these guys off could be leading to such debilitating conditions, what we're seeing with long COVID, potentially, and then post-vaccination syndrome, and then as well as cancers take advantage of that. So you can see the mitochondria in pig. Well, I was surprised to see how little cytoplasm there was. So cytoplasm was, I think, in green. This is from memory. I hope I didn't screw up the colors, but green, there's barely any liquid left. Cytoplasm is the liquid inside the cell. The organelles take up so much space, there's barely any liquid, room for liquid. And then there's, of course, the membrane itself around. So they use this technique just to show how powerful it is. But they measured something else. they specifically focus in this study to look at cytochrome C. Now, what is cytochrome C? It's a protein inside mitochondria that is absolutely necessary for burning fat and creating ATP or sugar and creating ATP molecules out of that process. ATP, think of ATP molecules, are the energy source that we use. So mitochondria are huge in producing energy for you. And you need cytochrome C because cytochrome C inside, it has iron molecule. And that iron molecule, that iron atom can capture electrons. So it can shuffle electrons inside mitochondria from one molecule to another. And this shuffling of electrons is what creates an electron transport chain. And that is what is absolutely required to produce energy. All right, so that's the bit of a background. Now, when the cytochrome C releases electron or no longer has an electron and it's ready to accept an electron, that is referred to as it's being oxidized. So it's an oxidized cytochrome C, and that iron molecule is more positive, all right? And then if it accepts electron and moves it down the chain, when it accepts that electron, it's reduced, and that iron molecule is less positive, right? So you, as I mentioned, you absolutely need this. And when mitochondria becomes dysfunctional and cannot do this anymore, what mitochondria will do, it will release that cytochrome C into the cytoplasm. The cell can detect that, and the cell can literally commit suicide because it's like, oh, my mitochondria are not working properly. This is not good news. All right? So you need that cytochrome C. And for these two purposes, check out my past videos dedicated to just mitochondria, how powerful these organelles are for our biology. They're doing far more than just create energy for us. All right, so then that's the bit of a background. Now you know what oxidized cytochrome C is. What these guys were using Raman for is to detect specifically that oxidized cytochrome C. And what they noticed is that when they expose these cancer cell lines to Pfizer mRNA vaccine, there was a reduction of that oxidized cytochrome C. And what does that mean? Is that there was less ability for now mitochondria to move that electron in its electron transport chain and burn fuel, which means less ATP being produced. That means these exposure to these vaccines happen to help the cancer cell line switch even more rapidly towards the system of burning fuel that you can see in some of the cancer cells that is anaerobic, meaning without oxygen. That's where you burn glucose without the need of mitochondria, because you're literally are compromising these, looks like these vaccines were compromising the ability of mitochondria to properly function. As a consequence, because there was also reduced levels of cytochrome C, oxidized cytochrome C, it also means that these defunct mitochondria were not able to release it into the cytoplasm and warn the cell, whoa, better kill me because this is not good. And they were also able to see reduced levels of apoptosis, which is a form of self-suicide that a cancer or any cell can commit. So these were two outcomes, and it was quite immediate. They were able to see it for number of hours, lasted for a number of hours that amounted to, I can't remember, maybe a couple of days or so. But then after 2 weeks, it leveled off and it finished. So in essence, to me, the way I see it, because I've been discussing this whole metabolic component, how cancer cells can take advantage of how they burn sugar to the area where they burn sugar without oxygen, and that makes mitochondria not work properly. This happens when you don't have either enough oxygen, so hypoxia, mitochondria are not working properly, or cancer cell lines, or now we're discovering potentially long COVID and post-vaccine injured people, post-vaccination syndrome individuals. So I thought I would bring it up together. All right, so this is gonna wrap it up here. So that tells you how it all tied together. Listen, guys, if you like this type of information, please subscribe. Hey, because it seems like there's a great story in every pixel you look at under the microscope. And give us a comment, leave a comment. And please hit that like button if you want, and subscribe if you want more of this type of information in the future, where we're learning crazy biology. This is constantly new information that I'm learning, and I'm trying to learn with you. And also, please check out my Patreon account, where I have additional content that does not make it to the YouTube. My patrons then vote and determine what content is good enough for YouTube versus what might be controversial or too controversial for YouTube, and therefore stays on Patreon. But I also have a lot of free content on Patreon as well, so subscribe. You don't need to be financially supporting member. And I look forward to seeing you on another trail. And remember, get out there, do lots of hikes so you can participate, have your mitochondria participate in oxidative phosphorylation and burn your energy fuel sources properly. Bye for now, everyone.